Hello, everyone, and welcome. Thank you for joining us today for our webinar, which is entitled Enzymatic Precision, Advancing Complex Small Molecule Synthesis Using Biocatalysis. I'm Masha Karanov, Director of Business Development for North America at Enzymatic, and I will be the moderator today. I'm excited to kick things off by introducing Enzymatic, a platform-based contract research development and manufacturing organization, also known as CRDMO, dedicated to accelerating complex drug substance development. At Enzymatic, we believe a platform-driven approach is essential to delivering agility, flexibility, and most importantly, predictable results. By integrating chemistry and biology, we support programs from early design and pathway development to process optimization and manufacturing. This enables the development of complex drug substances, helping bring life-changing medicines to patients more efficiently. Today, for the purpose of this webinar, we will be focusing on the biocatalysis and exploring core concepts behind the use of enzymes as catalysts and their expanding role in modern chemistry and bioprocessing. I'm pleased to introduce our webinar speakers or participants. We organized this webinar more of a discussion than a presentation. And I would like to start with our invited guest, Jim Bruno. He is the owner of Chemical and Pharmaceutical Solutions and consulting firm and former president of DCAT. With over 4 decades of pharmaceutical experience, Jim has been consulting since 2002, specializing in API and dosage form development for emerging pharma companies. And another very important speaker today is Ruben Carr, the head of chemical biology at Enzymatic. With more than 25 years of experience, Ruben has led the creation of numerous proprietary biomanufacturing processes for high-value therapeutic small molecules and intermediates. His work emphasizes the pioneering integration of chemoenzymatic methods to deliver scalable, cost-effective solutions. To begin, I think Jim will share his perspective on benefits and limitations of biocatalysis, drawing on his extensive experience in process development, industrial implementation, and then Ruben will join that discussion afterwards. So Jim, please, you have a floor.
Thank you. Thank you for the introduction. So I deal a lot with the products that are coming out of early development. So you see technology as they are doing it. And you can see a shift in the technology. I mean, years ago, when continuous processing was being adapted, it took a while for especially the pharmaceutical markets to adapt to those. When you look at some of the chromatography methods, you see people now using a lot of chromatography for different processes. Molecules become more complex, and these technologies are implemented. Biocatalysis is exactly the same. It's not a new technology. It's been around for years, but like all of technologies, it has its place. It's being used a lot. Companies and scientists are looking at these types of processes for a number of reasons. But at the end of the day, there's things that you have to be concerned about. And I don't care if it's a continuous process or it's a biocatalytic transformation. These are the types of things that in many cases you need to be concerned and considering, if you will. And in some cases, they're perceived to be a problem, and in some cases, they really are. I mean, consider biocatalysis. They're done in water. So you could have limited operating parameters that you can work with. A lot of biocatalysis also are not stable in various pHs, so your pH range may change. Your products may not be stable in water. Water sounds like a great material to use, but getting rid of water is a high energy cost. And when you're looking at the cost of these materials, your waste streams are becoming more critical when it comes to the cost associated with it. And I'll talk about waste in just a second also. So we have to be concerned about not just the beginning and the operation, but also at the end of it as well. There is still, I think, and some people say the same, a designability and stability of the biocatalyst. you have to be concerned with the stability of a catalyst, like anyone. And also, when you're in it, you may lose the ability to do the transformation that you need done. The conditions for your reaction may have to be so stringent to maintain that efficiency of the stability. This could be a concern, especially as the chemistry goes even larger and you're trying to scale up the manufacturing from grams to kilos to hundreds of kilos to tons in that regards. Also, designability, it's a little more difficult often to design a catalyst that will work very well in your system. There are catalysts that do basic transformations. They do a great job at that, but sometimes to do it for your system, it may not be the exact one that you need. And then you need something that has to be done to modify or change the catalyst. That could take time. There are companies out there, this webinar is an example, and Ingenza is an example. They can modify their catalyst, and therefore by working together, you can get these kinds of changes more rapidly than we could years ago. There are some public hurdles that you have to deal with. This whole idea of genetically modified materials, whether they're their catalyst or whether they're in your food to begin with, this hits the press, it's a problem. Keep in mind, you've got a water system. If these get into the mainstream and they're consumed by either animals or people, it causes a red flag to go up. So you got to be cognizant of that and what's going to happen at the very end of your process, not just at the beginning of it as you move forward. Again, looking at it as a scale up. What we do in the lab is one thing, but what I do in manufacturing may be totally different. And I got to be aware of that going forward. There are regulatory hurdles that you need to be concerned about. Like everything else, your impurity profiles, your catalyst, they may have side reactions. This could be a problem like any chemical reaction. We're getting better with our analytical methods so we can measure these things. So as we are scaling up, we can see the impurities, we know what's going on, the side reactions, the degradation products, which could react again and form something else from the catalytic transformation. So like all of the chemical reactions moving forward, especially in a pharmaceutical in application or some of the specialty chemical ones, these types of things have got to be looked at and then looked at very carefully. So whether if it's in the US FDA or TSCA, or it's EMA and REACH in Europe, we've got to be aware of what's going on and how are we going to deal with it from a regulatory environment as well. I know from the FDA's point of view, they're getting much better with this. They've formed a group for new technologies. They've got guidelines for emerging technologies. So you have people there that you can talk to. They give seminars showing these are what we're going to look at. And these seminars are talks by the people who are going to be reviewing your documents. So from that perspective, it's a lot easier to do that because you know what you're going to be looking at in the future. Things will change, but you're going to have a good basis for that. The waste stream complexity, I think, is also a major concern that we have to deal with. When you get done, these biocatalysts are going to go into your waste stream. You're going to have to treat them. It could mean dealing with your waste stream is a much more difficult problem to deal with. You've got materials in there which you're using to stabilize the waste stream and to modify the waste stream. How are they going to affect the catalysis, the biocatalysis? How are they going to be able to remove or destroyed? And again, when you're looking at it, water is expensive. You may not be able to recycle it. You may not be able to reuse it. So therefore, you're going to have to be cognizant of the fact that this is an added cost to it. So that, this is something you need to be watching. Like any chemical reaction, you're going to have a problem with the formation of unwanted byproducts. Where are they going to come from? Unreacted reagents, the impurities in your unreacted reagents, side reactions, and that type of thing. Reactions of the side reactions. These are all things that we're going to have to be looking at, just like every other chemical reaction. The biocatalyst, is it going to react with one of my side products? Is it going to react to one of the other places on a ring structure or on a long chain? And so if that happens, How am I going to get rid of it? What kind of processes am I going to have to be putting into the thing? So again, these add cost and they add time. I tell people when you're looking at a process, keep in mind that every time you touch it, you're going to lose your final product. And if you do simple math, take a multi-step process and get a 90% yield, do it for several steps, and you'll see how low your yield is after multiple steps, even though you think your step is really good. It's about the total process at the end of the day. So these unwanted side products are a concern, and these are things we need to be able to look at, and we need to be able to look at them earlier on in the process. So when I'm working with an emerging company in particular, and I'm looking at processes, and I'm looking at scaling them up to the next level, these are some of the areas that I'm looking at and my concerns, and the concerns that my clients have for how do I get to the next level? How do I take it from my lab, which was done in beakers, to now in large round bottoms, to small reactors, and then into a final commercialization? So, like everything else, these are the things you need to overcome.
Oh, thanks, Jim. That was great. I think you laid out the challenges and of what biocatalysis needs to deliver against. Perhaps to respond to some of the challenges you've laid out there, I want to give you some of the case studies and success stories we've had at Enzymatic, where we successfully implemented biocatalysis to address some of those specific challenges you've identified and described there to us. When we look at medicinal chemistry through discovery and preclinical, what you're seeing there, we've found is typically synthetic chemistry is the predominant methodology that's implemented. But the downsides there, particularly with regulatory challenges, and you mentioned there regulatory hurdles, is chirality and the necessity for chiral pharmaceutical products from a regulatory perspective that they be in excess of 99% in enzymatic excess is the typical benchmark regulators require pharmaceutical products to be prepared at. So, and some of the analysis we've done with emerging companies in the US in particular is synthetic chemistry is absolutely used as the predominant methodology in the discovery preclinical work. And how they address chirality is typically leaning on the use and implementation of chiral chromatography to separate a racemate product, which is what synthetic chemistry typically provides, into the single stereoforms for the preclinical and onwards through the pipeline drug development activities. The downside with that, though, is the cost. Cost to implement that stage in that route manufacturing is typically high. The chiral columns are expensive, using a lot of mobile phase to do the elution. You then have your suring effectively half of your material if you have a non-selective synthesis. So that overall negatively impacts both the cost, but also the process mass intensity of the manufacturing route. So that's an area where I think we've got where biocatalysis can really stand out and offer new opportunity to improve the toolbox of what synthetic chemistry can offer. And hopefully this case study I've got here exemplifies exactly what I'm describing there. So this useful chiral synthon here, D-tyrosine, how might a synthetic chemist access that? You'd need to think about protecting groups, probably, transition metal catalyst, probably, and the likelihood of that being able to achieve greater than 99% EE off the bat is probably low. In our case, we successfully implemented a one-pot process using two coupled enzymes, so specifically this L amino acid deaminase and D amino acid transaminase. So the first transformation, taking L-tyrosine, so L-tyrosine is a proteogenic amino acid, is made industrially at large tonnage volume today. So the cost position of this is low and it's readily accessible. Through the deaminase, deaminate it to this keto form, and then through a transaminase, we're using an amino donor, D-L-alanine, using the D form of that, we can successfully convert the keto into the D-stereoisomer. And at Enzymatic, we were able to do this as a one-pot process, accessing D-tyrosine from its L form with a greater than 70% chemical isolated yield and achieving greater than 99% EE in the process. So I think if you look at that as an operating process, it'll be difficult for synthetic chemistry to achieve stereo inversion in a one-pot process under these mild operating conditions.
Ruben, you make some really good points. And I think when I'm looking at manufacturing, there's a couple of things that really draw to what could be important. So the first thing, if you go to a one-pot synthesis, it eliminates some of my extra cleaning steps. And again, all of that goes into a waste stream. It's all material that has to be eliminated. Plus, it's time. Reactors that aren't making products are losing money. I mean, a manufacturing company, they want to keep those things generating something that's positive. Cleaning them is not positive. It's something you have to do. So I think that's a critical issue that is very beneficial. Also, timing. You know, the more, if I have a reactor and I can do something with it and it only takes a day as opposed to 2 or 3 days, that allows me to do two things. Maybe it allows me to increase my capacity without necessarily increasing my capex. So all these are benefits, maybe not on day one, but you know, for people that are looking at the development of a product going forward, these could be very big economic improvements in the process. I think the other thing, when I look at slides like this and I talk to people like you guys, you know, I looked at biocatalysis in the beginning as a way of getting chiral molecules. But even if I look at your first step, that, you know, that first step is making a non-chiral molecule. So my point is, you know, it's like everything else. These are not just for chirality. There's a lot of chemistry that we can do around them also that brings value to what we're doing. Your high yield is essential in that regards. The higher the yield, the more material I'm making, the less waste, and it becomes more valuable in the long term, and it affects the economics of my entire process.
Great. Now, thank you, Jim. I think perhaps another area we've identified where we think biocatalysis can really offer some opportunities in the toolbox for synthetic methodology is in regio control. And the next case study I want to share with you, I think illustrates that quite nicely. Where in this assignment we were asked to work on some time ago, considered this starter material, which has 4 sites of potential glycosylation, and the customer required specifically the 2 glycosylated products. So you then pose that question to a synthetic chemist: how might that be accomplished when you've got potential 4 sites of reactivity? You're looking towards the protecting, deprotecting group strategy to accomplish that control. What we implemented at Engenza, we took a different approach. We use an enzymatic approach using a glycosyl transferase class of enzyme, and using inexpensive glucose donor substrate, we can affect stereo-selective 2 control at the 2 position in a one-pot process in water at mild temperature. This is so we're not having to do any protection, deprotection. So in terms of the process mass intensity, the cost of goods to manufacture, and the lack of requirement to do protections, transformation, and deprotections, we avoid all of that complication. So we're able to achieve a very selective, controlled reaction in ways that I don't believe synthetic chemistry could achieve without use of an enzyme here. So
yeah, and again, I think your point is well taken. And again, from another side of it too, is we have to be careful with some of these chemistries. From a regulatory point of view, I've worked on several projects where the unwanted compounds, these impurities, are very hard to remove, and they could become a tox issue. On one drug that I worked on, it was critical to remove that, and the chemistry became extremely complicated because that impurity was toxic. And when you get a higher dose product, toxicity becomes an issue. So, you know, these are things that you may not know in the beginning when you're designing your chemistry, but these are things we've got to be cognizant of. And again, you're showing an example where I don't have to worry about those things. The other thing, you talk about deprotection, a lot of those are done with some kind of a metal catalyst. Now, grant you, a lot of deprotections are low pressure, so maybe it's not a special reactor, but it's still a metal. The specs for palladium and platinum in these reactions, which are commonly used, that continues to drop. And the regulatory environment around it says, I can't have 100 ppms of palladium or platinum in it. I work with a lot of processes now that are well under 10 ppms, and we're using technologies to get it well under 10 ppms or whatever in that regards. And again, those things affect your overall cost. I may always be talking about cost, but no matter what anyone tells you, at the end of the day, it's going to be about the cost of your drug. And that could be one that makes a difference. And doing this type of thing, I could envision, could significantly change the product. It's going to change the timing and how much I can make and the overall cost of it.
Yeah, thanks, Jim. Perhaps just to put one more case study up here is just how we go, our approach to how we examine a customer's target, and we take a retro-biosynthetic analysis. So synthetic chemistry, be familiar with the retrosynthetic analysis. We take that, but we broaden it and include use of enzymes in that retrosynthetic analysis. So in this specific example, we had a client come to us where they had an entirely synthetic chemical route to their target, including 10 chemical steps with multiple protection deprotections within it. And we had a look at that molecule, and through our retro-biosynthetic analysis, we're able to propose 2 alternative routes, implementing use of synthetic chemistry, but also biocatalytic transformations. So on this option 1, use of a proline hydroxylase, and on option 2, a threonine aldolase. So, and this also provides opportunities for redundancy and having through the feasibility work, mitigating risk of failure of any one of those 2 enzymes failing in the initial assessment work. So this, going back to some of the points you made earlier, Jim, we were able to cut down the number of steps in the operating route from 10 down to 3, using 1 enzyme and 2 chemical transformations, and simplify their overall process to manufacture this type of product.
Well, and again, I think you mentioned the word risk, but think of your risk also from the other side of it. How often when you're getting up to a larger scale, you may not do it to late phase 2, early phase 3, but you start looking at a second supplier. And the technology and the chemistry that you're offering is not something that I can't take someplace else, or maybe somebody else develops a similar path but uses a different enzyme but can get the same results. It allows me to have more flexibility in that process, and I'm not limited to a specific product area or country or company in that regards. And that will de-risk my process significantly. And again, from the emerging company's point of view, who is at that point trying to sell the asset, these bring added value to the assets that they're trying to sell. And so you're scaling up, you're using standard reactors. So again, I'm not really looking at a lot of new CapEx, which helps me on the long run. I can do it in different ways. It may still be a biocatalytic transformation, but what I developed in phase 1, by the time you get to phase 3, it may be on the 3rd iteration of that catalyst in that regards. And I think those also are key drivers for what I'm looking at going forward. So you introduce a lot of those kind of opportunities, and again, you've reduced steps, you've reduced cleaning, you've reduced waste, in that regards. And side product problems, I don't care how you do it, even with all the blocking groups and deblocking, you're still going to have problems there. You're going to have to watch them. And if I minimize the steps, I also potentially minimize some of the impurities I generate, and therefore I may not have to clean them up at the end of them. So there's a lot of value in how you're going and what you're doing. And these are things that should be looked at during the entire chemical pharmaceutical process, if you will, or any chemical process that you're doing.
Yeah, and one of the questions often posed in this type of challenge of integrating biocatalysis within synthetic chemistry is, well, how do we get the enzymes? So I perhaps want to finish up with some of the case studies and where we've achieved successes in using our technology, platform technologies, to enable rapid, low-cost production supply of the enzymes in question here. So specifically, I want to introduce you towards our ingenious host platform. Classically, most enzymes will be made through E. coli, typically, which is useful to make intracellular biocatalysts, but we also have some additional options with Corynebacterium glutamicum, soon known as Putida, which can give new opportunities in enzyme improvement for production. We also have a repertoire of different hosts which enable the enzyme to be produced outside of the microbe producing system. So as an extracellular biocatalyst production, notably in yeast systems such as Saccharomyces cerevisiae and Pichia-Pasteurus, also other bacteria such as Bacillus subtilis, and fungal systems such as Aspergillus niger. So, coming to Ingenza, working with us, it's not just about, well, we can get your biocatalytic process to make your product. We can also support the enzyme production part of that journey as well. In addition to the host platform we also have our technology called Codable, which is a machine learning algorithm which allows us to exploit the redundancy in the genetic codes of the DNA which encodes enzymes to benefit and maximize expression yield, which overall lowers the cost and cost contribution of the enzyme to implement its use in the manufacturing process. So here we can see the first lane here on the left-hand side is the molecular weight ladder, the control, so the host system without expressing the respective enzyme. Some gene synthesis providers which have an optimized codon algorithm of their own and our own proprietary method. And you can see the fatness of the band here reflects the intensity of which we've improved the enzyme expression and thus lowered the overall cost to produce that enzyme. And that addresses how much enzyme ways we can make it. You raised the point earlier of how can we change the catalyst to make it work on the specific compounds. We've also got strong experience in direct evolution. So direct evolution is the principle where we can take some very low activity and turn that dial right up to increase the catalytic efficiency of that enzyme. So in this specific example here, WT, the wild type activity, there is activity on the given substrate, but it's pretty pathetic, to be honest. And the first generation, there was a little bit of improvement. Second, third, and fourth generation, though, we got some real gear changes, improvement in productivity, and we got over four orders of magnitude improvement in catalytic efficiency. And we're able to implement the use of this enzyme variant in the deracemization process here. So taking a racemic amine and making a completely stereoselectively stereoconfigured amine in one pot process using this biocatalyst. So using the combination of how we produce the enzyme, but how we can tailor the enzyme enables us to really address how we can meet the challenge of making clients' specific transformations work for them at the cost position they want. So I think we're probably done there, Matthew, I think.
No, I just wanted to say, well, excellent. I think in the interest of time, we should be wrapping up, but I'd like to summarize or conclude, if you will, with the question to both of you, and that is, I'm hearing that biocatalysis serves as a complementary approach to synthetic chemistry, and in many cases can be a powerful tool to overcome some of the inherited challenges in synthetic chemistry. Am I correct in this conclusion, right? Is that what I've heard from both of you?
I think even to that point, I think because they've been shown to have some extremely good value, and a lot of the problems that we may have addressed 10 years ago, 20 years ago, whatever, they're being overcome. And now it's just a regular method that we're using. I mean, I hate to use the word in a sense, but it's not special anymore. It's just like every other reagent that we've ever used. And again, it's a tool. The chemist gets it, they get used to it, and there's enough history on it now that says, yeah, I can do that. It's not a big deal, in that regards. And that's where you want to be. You want to take that fanciness out of it, so to speak. And then, oh my God, what's that all about? Well, that's gone, in that respect. Now it's a catalytic transformation. Yeah, no problem. We can do that.
Excellent. Ruben, your thought?
Yeah, no, I agree with Jim, and I hope that trend continues, a wider and increased adoption of biocatalysis in medicinal chemistry. Yeah, and I hope people consider working with the use of biocatalysis to help support their own product needs and exploit the tools and the platform technologies we have to expedite their own product manufacture.
Excellent. So with that, I would like to extend sincere thanks to both of you for insightful discussion. I've learned a lot. And due to time constraints, we won't be able to address questions live, but rest assured, we'll be happy to follow up with you after the webinar. So you'll receive notification once the webcast is available for replay and download. And with that, we will conclude our webinar. And once again, thanks for joining us today, and enjoy the rest of your day. Thank you.
Thanks.